Welcome to channel Conceptual Biostudy. My dear students, if you like my video, please subscribe it and share it with your friends and others. Thank you all. My dear students, today we will study about Pteridophyta, that is third division under plant kingdom. The term Pteridophyta is coined by scientist Haeckel. Study of Pteridophyta is called pteridology. Pteridophyta is commonly called number 1 vascular cryptogams. Vascular cryptogams. Vascular cryptogams. Number 2, they are also called amphibian of plant kingdom. Amphibian of plant kingdom, like Bryophyte. Number 3, they are called botanical snake. They also called reptile of the plant kingdom. Reptile of plant kingdom. Okay. Next, about their habitat. They are mostly terrestrial in nature. They are mostly terrestrial in nature. Terrestrial, that is, live in moist, cool, shady places. Mainly in soil along roadside or railway track or on moist old wall or on moist tree trunk. Okay. Sometimes they are also found on rocky surface, in cold region. Okay. They may be aquatic. Number 2, these are the first habitat. Number 2, they may be aquatic. Like Marsillia, Salvinia, Azolla, Isotis. Okay, Isotis. Number 3, they may be epiphytic in nature. Epiphytic. Example, ek number, Lycopodium phlegmaria. Lycopodium phlegmaria. Lycopodium and Osmunda. Number 2, Osmunda pendulum. Due to pendant in nature, that is the stem hang downwards. Next point about their special features. Look here, next topic, special features. Special features. Number one, they are fast cryptograms. They are fast cryptograms. Look here, actually from thallophyte we have seen that they are also cryptograms, bryophyte also cryptograms, but here fast cryptograms which possess vascular bundle. Fast cryptograms which possess vascular bundle, which possess vascular bundle. Okay, vascular bundle consists of xylem and phloem. Look here, vascular bundle consists of xylem and phloem, but they are primitive type xylem. As initially grow that here for the first time vascular bundles are present here, so they are primitive in nature. Why primitive in nature? Because no vessel or trachea in xylem. Okay, xylem, and no companion cell in phloem. Okay, so these are cryptograms and vascular cryptograms. Okay, next point, cambium is absent here. Next, cambium is absent here. So no secondary growth. But exceptionally in isotis, aquatic fern, already I have told you, isotis, secondary growth found. Okay, next point, vascular bundle, number one, concentric, not conjoined, concentric. That means one tissue is surrounded by another tissue. Here they are heterocentric, that is centrally located xylem and peripherally located phloem. So they are amphictyval, concentric, and as amphictyval or hydrocentric, as hydro means water, that is water carrying tissue in the center, so it is called hydrocentric or amphictyval. That is amphi means phloem, phloem means phloem surrounded. Hydrocentric or amphictyval. Okay, and open and closed due to lack of cambium, and closed type, that is closed type due to lack of cambium. Okay, and mesarc, and xylem is mesarc type, that is protoxylem more or less surrounded by metaxylem. Next point, they are successful land plant. They are fast successful land plant. Okay, look here, actually or in bryophyte class I have told you that bryophyte is a fast land plant but not successful, but here it is first successful land plant because due to presence of vascular tissue. Number 1, vascular tissue, and number 2, presence of root. But look here, they are not completely successful land plant, but they are not completely successful land plant. They are not completely successful land plant because they are first successful land plant, but completely, number 3 now not completely successful, completely successful land plant, because they need water, to accomplish fertilization. So their gametophyte need damp and shady place. So that is to survive their life, that is especially their gametophytic body need damp and cold or shady places. They especially gametophyte need a cool, damp, shady place. Okay. Next point. Only group where, number 4, it is only group where independent alternate life cycle is found here. Independent alternate life cycle is found here because here gametophyte and sporophyte are morphologically different and separate, and sporophyte that is haploid and sporophyte that is diploid. Sporophyte are separate and one does not depend another one. One does not depend on another one. That means each of them are independent and carry out photosynthesis. You know that the gametophyte, look here, this is the diagram of prothallus, that is gametophyte of Pteridophyta. It is green, and I have this is why I have gave green here. It is photosynthetic. So both of them are independent. Okay, both of them are independent. Next, it is the last group. Next point 5. It is the last group where antheridia is present. It is the last group. It is the last group 5. It is the last group where antheridia is present. Where antheridia, that is male sex organ, antheridia is present. Please mind it that from the next group, that is gymnosperm, angiosperm, you will never find antheridia. Antheridia is here for the last time. Actually, it developed in Thallophyta, especially in algae, class algae. Okay, so it is a special character. Now, next point, anthrozoid. Next point about anthrozoid. Next point about anthrozoid. Anthrozoid, that is sperm. Anthrozoid may be commonly spiral and biflagellate mainly, and multiflagellate, and mainly multiflagellate. It is found in example Xylotam, very primitive genus, but there spiral shaped anthrozoid and multiflagellate Xylotam, and all members of funds that is Phrygopsida funds. Okay, look like this. This is spiral shaped flagella and many flagella. Okay, many flagella. This is anthrozoid, or maybe spindle shaped and biflagellate. As in Selaginella and Lycopodium. Example Selaginella and Lycopodium. Okay, Lycopodium. Look like this is spindle shaped and this is biflagellate. This is look like. Okay, next about in their life cycle, both, very important point, in their life cycle both rhizoid and root are present. Rhizoid and root are present. Look here, rhizoid is present in Xylotam. That is Xylotam, primitive type of further discuss rhizoid in Xylotam, where root absent, rhizoid present, rhizoid present. But in another genera, rhizoid is also present, but they are present in their prothallus or gametophyte. Generally gametophyte is called prothallus, though in Salvinia, Selaginella, Marsilia, they are not called prothallus. Look here, this is the prothallus, this is the rhizoid, this is rhizoid. So in their life cycle, this is the gametophytic stage, they possess rhizoid, and in some cases like Xylotam, this is rhizoid present in sporophytic body. Look here, in sporophytic body. It indicates sporophytic body. It indicates that it just arises from bryophyte. Okay, arises from bryophyte. Next, about their spores, or on the basis of spores, they are called homosporous or heterosporous. Look here, next point 7. If we study the type of spores in the sporangia, next point, sporangia produces either one type of spores, or two types of spores, two types of spores. When one types of spores, they are called homospores. And when two types of spores, they may be macrospores or microspores. Macrospores, that is large, comparatively large, or microspores, comparatively small. Okay, and most of the pteridophyte, and from this point of view, most of the pteridophyte most of pteridophyte are homosporous, look here, homosporous. And you can recall that Biostudy, and if you recall that Biostudy, they all are homosporous, but here most of the Pteridophyte are homosporous, some are heterosporous, like Selaginella, Marsilia, Azolla, Salvinia, Isotis. These are heterosporous. That is produce two types of spores in their life cycle. Okay, and next point about fertilization. Look here, about fertilization. Fertilization is judeogamy. Already I have discussed this term in Biostudy class, that where the male gamete may be multiflagellate or biflagellate, they go from antheridia to the archegonia. Here archegonia, look here, archegonia possess one large female gamete, that is egg, and one ventral canal cell, and generally two neck canal cells. This is ventral canal cell. Look here, unlike Biostudy, here ventral canal cell also get destroyed before fertilization. Okay, and because here in Biostudy, especially in mosses, the ventral canal cells, I have told you that take part in the calyptra formation, but here there is no role, and here number of neck canal cells is short. When we proceed, the number of neck canal cells gets reduced. Okay, and when we reach to angiosperm, there we will found no archegonia. Okay, and also in Neet, being gymnosperm, no archegonia is found. So absence of archegonia or reduction of archegonia is the advanced character. Okay, let us come to the point. That is sperm or anthrozoid through water, it ultimately reach to the egg, and this journey of anthrozoid through water is called judeogamy. Okay, it is called judeogamy. So fertilization is judeogamy. But another characteristic feature, though found in a special genus, that is in Selaginella. In Selaginella, a special character, Selaginella, a special and advanced character is found, that is the development of gametophyte from spore within the archegonia takes place when the archegonia retained on the sporophytic body. Look here, retained on the sporophytic body, which is also happened in gymnosperm higher group, where archegonia, the ovule possesses archegonia, and within the archegonia sporophyte is developed for the next generation. But here also within the, suppose this is the gamete sporophytic body, this is the sporophytic body, and this is spike, and this spike or strobilus, and the strobilus means the lip, which sporangia, and when spores are formed, within the sporangia, these spores does not come outside. When the spores come outside from the sporangia and develop on the soil to form gametophyte, this is called exosporic germination. But here the germination of spore takes place within the sporangia to form female gametophyte, here female organ, that is archegonia, and within the archegonia the development of zygote, that is embryo is formed within the archegonia, where archegonia is still attached with the sporophytic body, and this is the characteristic feature, higher characteristic feature, and this indicate the precursor of the seed habit. That means this plant try to form seed, that is the embryo within the archegonia, that is within the female gametophyte, which is attached to the male, which is attached to the sporophytic body. Okay, so this is it shows seed habit, that is here development of embryo takes place, within archegonia, within gametophyte or archegonia, gametophyte still attached to the sporophyte, sporophyte. So it is seed habit. Okay, precursor of seed habit. It shows precursor of seed habit. Okay, so now about their sporophyll. Look here, sporophyll. Sporophyll means sporangia bearing leaf. So their sporophyll is important character is sporophyll is photosynthetic in nature. That is sporangia bearing leaf photosynthetic. Trophophyle also photosynthetic, but sporangia bearing leaf also can carry out photosynthesis, unlike gymnosperm. In gymnosperm we will see that the microsporophyll, megasporophyll, that is microsporangia bearing leaf or megasporangia bearing leaf, but they can't do photosynthesis. But here sporophyll can carry out photosynthesis. Next important character, sporophyll can carry out photosynthesis. Okay, photosynthesis. This is the last point and last special point. Okay, now we discuss about the life cycle in diagrammatic representation. Okay, diagrammatic representation of life cycle. Life cycle. First life cycle is diplohaplontic type. That is diploid stage and haploid stage are morphologically different and they are more or less same in duration. Diplohaplontic type. And you can also know that in diplohaplontic type of life cycle, there the characteristic feature of this cycle is sporic meiosis. That is, here meiosis takes place during spore formation from spore mother cell in the sporophytic body. This is the characteristic feature of diplohaplontic life cycle. And in haplontic life cycle, when I discussed about Thallophyta, I have already told that the zygotic meiosis is the characteristic feature of haplontic life cycle. And in Graphite also haplodiploidic life cycle, there also sporic meiosis is taken place here. But in gymnosperm and angiosperm, so at diploidic type of life cycle, their gametic meiosis is there. That is, during gamete formation, meiosis takes place. Okay. Now here diplohaplontic life, if we start from the main plant body, suppose it is main plant body. Obviously, it is diploid in nature. And main plant body, the important reproductive part, if we take, that is sporophyll. Okay, sporophyll, which is diploid in nature. Sporophyll, sporangia bearing. It produce sporangia. Already diploid. This sporangia produced by mitotic division, equational division, mitotic division produce spore mother cell SMC, spore mother cell, which is also diploid. This spore mother cell undergoes meiosis and produce spores. Already it is haploid in nature, and it is the first cell of the gametophytic generation. Okay, these spores on germination, look here, germination is of two types, exosporic or endosporic. Mostly exosporic, that is the spores after bursting of the sporangia, spores comes out and on the soil they germinate. This is called exosporic, but in Selaginella partially endosporic in nature. Okay, and in higher plant the spore germination is endosporic, but in lower plants the spore germination is exosporic. So on germination, they produce gametophyte. Look here, this gametophyte may be monoecious or dioecious. In case of monoecious species, spore produce gametophyte which is monoecious, that is the one single gametophyte produce antheridia and archegonia. But if the species is heterosporous, two types of spores and megaspores, that is megaspores produce female gametophyte and microspores produce male gametophyte. Whatever the nature of the gametophyte produce two types of sex organ, that is antheridia and archegonia. Antheridia male sex organ, antheridia and archegonia. Okay, antheridia and archegonia both are haploid in nature. Antheridia produces by mitotic division, obviously by mitotic haploid. By mitotic division produce so many anthrozoids which are in, and it produce egg which is haploid in nature. Okay, and these two by judeogamy type of fertilization produce zygote, diploid zygote, which on equational division and development produce the new sporophyte by growth produce main mature plant body, that is twice. In this way they complete their life cycle. Look here I have termed two special term, that is zygote and spore. This zygote and spore, very important, it is in spore is the first cell of the gametophyte. This is the twice, where the structures, where the stage which are diploid in structure, that is sporophytic generation. This is sporophytic phase or sporophytic generation or diploid generation. This is haploid stage or gametophytic generation. Okay, gametophytic generation. This spore is the first cell of the gametophyte. That is likewise zygote is the first cell of the sporophytic generation. And look here, this fertilization, another two term, fertilization and meiosis. Look here, this is the meiosis. Look here, these two term, meiosis is the very important, because this is the two opposite phenomena. Fertilization and meiosis is the opposite phenomena in an alternation of generation, because meiosis helps the plant from diploid stage to haploid stage, and it helps to haploid stage to diploid stage. So these two phenomena, that is fertilization and meiosis, is the alternate phenomena of a life cycle. Next, about general characteristics in short. Already I have discussed a lot. Now about their general features at a time. General features, number one, main plant body. That is sporophyte. General features in brief. Number one, main plant body, sporophyte. Main plant body, sporophyte, diploid, and it consists of mainly root, stem and leaf. Okay, root, stem and leaf. About root, I have already told that root may not be present in Xylota, that is under Xylopsida, what present, where root present, they are adventitious. They are adventitious. Though adventitious initially, when during the development of the young sporophyte, they are develop the primary root, but they are short-lived and get destroyed, and instead from the base of the stem there arise adventitious root. So mainly adventitious. About stem, stem is mainly rhizomatous. The rhizome like rhizomatous. It may be erect, it may be semi-erect, it may be semi-erect, or pendant, that is grows downward. Leaf, about leaf, they may be trophophyll, they may be scaly. First of scaly leaf, scaly reduced leaf in xylotam, primitive, that root. Look here, this is xylotam, this is leaf, scaly leaf, reduced. There is no vascular tissue here, and also rhizoid. In rhizoid also, this is rhizoid also no vascular tissue here. I forgot to mention a point, mention a vital point, that is gametophyte. In gametophyte there is no vascular tissue in any type of gametophyte. Either thallophyte, bryophyte, pteridophyte, gymnosperm, angiosperm, any type of gametophyte, there is no vascular bundle. Mind it. Scaly reduced leaves, example xylotam, very primitive genus xylotam, and only genus, this is living fossil. Other Rhinia, Horneophyton, they are extinct. And leaf, scaly leaf, and foliage leaf, which may be two types, maybe trophophyll, and maybe sporophyll. Look here, trophophylls mean vegetative in nature, mainly vegetative in nature, reproductive in nature. It is reproductive in nature. It may be present from the initial to the up to the end of the life, and they only carry out photosynthesis. And sometimes sporophyll, in some genera, some cells during the reproductive stage, they convert into sporophyll by bearing sporangia. And by bearing sporangia. Look here. This is trophophil, and as there is no sporangia. But if this leaf bears sporangia, look here, in this way, the leaf sporangia, then it is called sporophyll. Then it is called sporophyll. Sporophyll. That is sporophyll. But in some cases, like in ferns, look here. This is the fern. Look here. This is fern. This is fern. All small sporangia are developed here on the abaxi. No trophophil here. No trophophil. But when the young leaf, young stage, the leaflet or leaf is called trophophil, because they in their initial stage carry out photosynthesis, but mature stage, reproductive stage, all leaves becomes converted into sporophyll, which is absent. Sporophyll. Look here. This is another fern. This is another fern. Here also, and here another phenomena, heterophyll is found here. Look here, fern, heterophyll. This is the young leaf, and this is the mature leaf, highly dissected, but here the leaf is intact and other shape. So different type of leaves in the same plant, heterophyll, when you will learn about growth chapter, plant growth regulator chapter. Okay, and this is the trophophil. This is the trophophil. Okay, this is the trophophil, that it from the very beginning up to the end of the life, they persist in this way and perform vegetative functions. But the special leaf, look here, this is the strobilus, and this strobilus consists of trophophil. Okay, consists of trophophil. Now about their leaves, and this is the sporophyll. Sometimes sporophyll may be present in single form. Look here. Look here, this is the one type of draft trees. These are sporophyll. All the look. These are the sporangia, and this is the all are sporophyll. So here sporophyll never form a compact structure, and in this way they sustain their life. And sometimes they form a compact structure. Sometimes sporophylls forms a compact structure. Look like this compact cone-like structure, compact cone-like structure. This cone-like structure is called strobilus or cone. This is called strobilus or cone. Strobilus or cone. Strobilus. Look here, this is the strobilus of Lycopodium. This is strobilus of Selaginella. This is strobilus of Selaginella. This is strobilus of Equigetum. This is strobilus of Equigetum. So in most of the fern, though strobilus is present, but in true fern, most of the pteridophyte, though strobilus is present, but in true fern there is no strobilus. That is, each and every leaf at their reproductive stage becomes converted into sporophyll. Okay, so sporophyll, these are about sporophyll. Now leaves, about leaf. Another point. On the basis of their size, there are two types. On the leaf also. This is the tropophyll, sporophyll on the basis of their presence or absence of sporangia. But leaf, according to the size, they are two type. That is microphyll. And sporo and macrophyll. Okay, macrophyll. Microphyll means small in size, and uninerved. That is uninerved. Look here, this is uninerved. Only one midrib is present. Vein absent. Midrib is present, but is macrophyll. It is large. First of all, it is large and with midrib and vein. Look here, this is macrophyll. This is the macrophyll. This is a large leaf and stem is small in draft trees, and each leaflet bears a this is the midrib, and midrib is further divided to form a pinnate compound leaf, and midrib also possesses vein. These are the veins, so it is macrophyll. Microphyll example Lycopodium selaginella. Look here, this is the example of microphyll. The leaf stem is large and leaf is small, and in selaginella also and selaginella equigetum, these are look here, the stem in selaginella leaf is small, very small, and stem is large, macrophyll. And on this basis, pteridophyte are of two groups, that is microphylus and megaphylus. On this basis, they are of two types, that is pteridophyte are of two types, that is macrophylus and microphylus. Okay, leaf pteridophyte two types. On this basis, two types, that is macrophylus and microphylus. Macrophylus and microphylus. Macrophylus means leaf large, phylus means leaf, and macro means larger leaves and smaller stem. Example macrophyll. These are the macrophyll. They look here, and stem large, draft trees. Most of the fern draft trees, adiantum, draft trees. Look here. This is a small species of draft trees. The stem is very small. Look here, the stem this is very small, and leaf is very large. Okay, leaf is very large. Here also one type of plant. There the stem is very small. This is the stem, rhizomata stem. Here rhizomata stem. This is adventitious root, and this is large leaf. So it is macrophylus in nature. Okay, macrophylus in nature. Next, microphylus. These are microphylus. Leaf small. Leaves smaller, and stem larger. This is stem larger. Example Lycopodium selaginella. Lycopodium selaginella. Okay, selaginella. These are about their stem, leaves, etc. And next about Sporophyll. Okay, now we discuss about sporangial development of sporangia. Now we discuss about development of sporangia. Its important characteristics here. Development of sporangia. About development of sporangia. Development of sporangia. Development of sporangia, two types. Number, leptosporangiate. On this group, the pteridophytes are divided into two groups, leptosporangiate and eusporangiate. Leptosporangiate and eusporangiate. Okay, eusporangiate. In leptosporangiate means when the sporangia develop from a single initial cell. Sporangia develop from single initial cell. And here sporangia develop from a group of initial cells. Okay, and leptosporangiate, all ferns are leptosporangiate and equigetum also leptosporangiate. Example of leptosporangiate, equigetum, and all ferns, not equigetum here, leptosporangiate, all ferns like Salvinia, Pteris, Drapteris, Adiantum is also called walking fern, Adiantum, and eusporangiate, Marsilia, equigetum, etc. Selaginella. Look here, eusporangiate type of development is advanced because it is found in higher plant. It is advanced type. It is advanced as it is found in higher plant. It is found in higher plant. Okay, so these are about their development of sporangia. Okay, and now we discuss about gametophytic body. In short, gametophytic body. About their gametophytic body, mainly in most cases gametophytic bodies, in most cases gametophytic bodies prothallus. Gametophyte is prothallus. Gametophyte is called prothallus, except Marsilia and Selaginella. Okay, I have already I forgot to say one thing more, that is sporangia. Though I have told you that sporangia are developed on the adaxial surface or abaxial surface. In case of fern, they are produced on the abaxial surface, but in Selaginella Lycopodium, it is the adaxial or ventral surface, that is upper surface. But sometimes they are produced in a special structure that is called sporocarp, which is found in Marsilia. Look here, this is the Marsilia, and here a special structure here special structure is formed, is called sporocarp, and during economic importance I will say that this starch, it contains starchy mucilage which is used in nut dough formation, that is by Australian people. Okay, so about Prothallus, Marsillia and Selaginella. Prothallus is thallus like structure, green, autotroph, bears rhizoid, bears multicellular, this is multicellular here, and it may be monoecious in case of homosporous species, and maybe heteroecious, that is dioecious in heterosporous species, and they possess and Archegonia and Anthridia and Archegonia, and okay, and in this way they after gametophyte maturation of the gametophyte, there happens fertilization through zooidogamy, and they form zygote, and zygote develops initially on this gametophyte, that is for the initial development they do not draw nutrition from here, and when root is formed they become independent. In this way they complete their life cycle. Now we discuss about their classification in short. Already we have discussed a lot about their classification. Okay, about their classification. Actually, pteridophyte is divided into four classes. Number one, Psilopsida. These are class rank. Psilopsida. Next, Lycopsida, Sphenopsida, and last of all Filicopsida or fern. Filicopsida or Pteropsida, generally called ferns. Okay, ferns. Just in a short, first Xylotum, example Xylotum, only living species. This is called living fossil also, living fossil, and extinct species, two extinct species, Rhynia guenvaugani and Horneophyton. These are extinct species. Horneophyton. About Lycopsida, example Lycopodium and Selaginella. Lycopodium and Selaginella, very interesting species. Selaginella. Sphenopsida, equisetum. It is also living fossil because two Sphenophyllum calamities. These are extinct species. Under this group, extinct calamities, Henia, etc. and all ferns, that is Adiantum, walking fern, Adiantum, Diapteris, Pteris, etc. Diapteris, Marsillia, Diapteris, Pteris, etc. Okay, and about Xylophyta, this is Xylophyta. Just look, this consists of rhizoid, no root, rhizome stem, and rhizomatous stem, and stem, they are branched. Stem is rhizomatous, and prostate part actually stem is dichotomously branched. Look here, dichotomously branched, vascular bundle only present in stem, because their root and leaf, leaf is scaly, stem and leaf, scaly leaf, scaly leaf. And about the sporangia, there's look here, the sporangia is produced on the axil of the scaly leaf. Axis sporangia present on or produced on axil of scaly leaf, and it is look here scaly leaves, and these are trad in nature. Trad, that is only one sporangia are produced 3 sporangial sac, and the spores are bilateral. Look here, these are the spores, bilateral symmetrical. And about the gametophyte, their gametophyte is cylindrical dichotomous. Gametophyte is look like this, gametophyte look like this. This is the dichotomous gametophyte, and they possess rhizoid here, and haphazardly distributed sporangia, antheridia and archegonia, and they produce antherozoid which are spiral and multiflagellate. And about their archegonia, they are short neck, possesses 2 neck canal cells and 1 ventral canal cell and 1 egg, and they perform sexual reproduction as usual. About Lycopsida, look here, Lycopsida, we don't like to write further. Lycopodium, Lycopsida, these are microphyllus, that is leaves are small, stem is large, and their body consists of stem, leaves and root. Root is adventitious, developed from the base of the stem, and leaves are very small. They are spirally arranged, sometimes in opposite direction, opposite phyllotaxy, and in whole phyllotaxy, sometimes and at that, and they are branch is dichotomous branch. Sometimes the distal branch, look here, this is the distal branch, not appears as monopodial in nature, that is they arise side by side, that is not far distance like that. And at the tip of the branch there arise some strobilus. Strobilus means the sporangia form a compact structure surrounding a central axis, but they are loosely arranged. These are about Lycopodium, and their gametophytic body produce autotroph, and exosporic development, germination is found there, and other characteristics like normal pteridophyte. Okay, about Selaginella, very interesting genus Selaginella. Here a special, it is also microphyllus, that is stem is large and leaf is small. Leaves, 2 types, main leaves, small leaves and large leaf. Large leaves so is ligulate, that is tongue-shaped leaves, and their roots adventitious, but a special structure is present above the root. Look here, this is the adventitious root, and above the root there is special structure rhizophore. It is also called organ of sui generis, that is morphologically to some extent like that of stem, like that of root. This is and another characteristic feature of Selaginella, that it can roll up, that is get uprooted, automatically uprooted in dry or drought season, and they roll up and form a brown colored ball like structure, and they tide over the dry condition, and when normal condition, that is favorable condition, they automatically absorb water and becomes flattened and behave and perform their normal functions. And besides this, already I have told you that it is another characteristic feature, that is development of the sporophyte within the gametophytic body, where the gametic body is still attached with the sporophytic body, that is it shows seed habit, that is precursor of seed habit. Okay, and now about Sphenopsida. Sphenopsida, equigetum, living fossil. Look here, this is the equigetum. Here 2 types of stem is rhizomatous, and they bear 2 type of branches. This is for sterile branches, and this is fertile branches. Look here, fertile branches form strobilus, and here the strobilus is developed within a special structure which is called sporangiophore. This is called sporangiophore. sporangiophore, and look here, their leaves are also small, very small leaves. Look at these are scaly leaves and cylindrical in nature, and they fuse side by side and form a sheath-like structure. Look here, a sheath is present already. I show you that this is original equisetum in dried form. I have collected it from Koushani, 6,200 feet in altitude. Look here, this is a scale-like this is a scale-like structures, leaves, and these are the lateral branches, and this is the vegetative thallus. There is no strobilus here, but here it is fertile branch that produce strobilus. Okay, and their roots are adventitious, and sometimes they look here, they produce tubers which acts as vegetative propagule. And about their gametophytic body, fertilization is all cases. Fertilization is judogamy and of course oogamous type, because the gametic fusion taken place between anthrozoid and spindle shaped anthrozoid or spiral shaped anthrozoids and ovum. Okay. And about Phelicopsida, this is the large group, and they are known as ferns. Already I have shown you some example original ferns, and this is the Diopteris. It is one of the best ferns, and you look that it consists of rhizomata stem which possess adventitious roots, and they sometimes ramenta. Look here. Though it is not so much visible, but young stem, look here, it is a characteristic feature. Look here, this is the young leaf, and the young leaf, apex of the young leaf remain coiled like the spring of the watch. This is called sarcinotixis. It is the very characteristic feature of fern. Another characteristic feature of fern, they are all turophyll, and on the sporangia are born on their abaxial surface, not adaxial surface like Lycopodium selaginella. Okay, and they do never form strobilus or cone. Okay, and next they show leptosporangia type of spore development, or the sporangia, that is sporangia develop from a single initial cell. Okay, and if we notice the sporangia, it is encircled by a jacket, and this jacket consists of two different types of cells. Look here, this is stomium, highly thickened wall stomium. This is stomium. Sorry, the thin wall is called stomium. Thin wall structure is called stomium, and the thick wall structure is annulus. It's called annulus, and these are this stomium is hygroscopic in nature and helps in spore dispersal. When it gets burst, the spores are come out. Generally, just a minute before I told you that Pteridophyte as a whole mostly homosporous, but most of the ferns are homosporous except Marsillia, Selaginella, not fern, Marsillia adianta, Marsillia salvinia, these are being fern, and they are heterosporous, Marsillia. Okay. And about their gametophytic body is prothallus. Look here, it is a prothallus of Diopteris, and it is autotrophic, green, thallus-like structure, 1 to many layer thick, and from the margin there arises the scales. These are scales. This helps to absorb water from the water and keep it moist. Okay, and between the rhizoidal branch, there arise the antheridia, and they possess a notch-like structure, surrounding notch. There arise archegonia. This archegonia is short-necked, and they produce multiflagellate sperm. It is another characteristic feature of fern is the multiflagellate sperm. So these are the characteristic features of all Pteridophyte. Now we discuss about the last topic, that is economic importance of Pteridophyte. Just as in a short look here, Pteridophyte, number one, about their economic importance. Economic importance. Number one, various species of Lycopodium. Look here, they are very nice in structure. Various species of Lycopodium, various species of Lycopodium, especially Lycopodium obscurum, one species Lycopodium obscurum is called Christmas green, and it is green used as garland in Christmas festival, and also in decorating purpose, due to its nice structure, decorating purpose. Okay, decorating purpose. Number 2, medicines are also produced from stems and pores of Lycopodium. Medicines also produced from stem and spores of Lycopodium. Okay. Number 2, spores being highly inflammable. Spores of Lycopodium being highly inflammable, they are used in theoretical performance during producing lighting, because it produces yellow colored light like the sulfur burning. So under the heading vegetable brimstone, vegetable brimstone. Okay, vegetable brimstone. Number 3, selaginal also used in decorative purpose, because selaginal is also look very nice. Also some species are also kept in one's garden. Also selaginal is also also as garland and also used as garland and decorative purpose, also used in decorative purpose in festivals, decorative purpose in festivals, and for its high conservancy, because it can hold water. Conservancy. Number 4, the underground modified stem of isotis and corm. Next point, the underground modified stem. Underground modified stem of isotis, that is corm, in the form of corm of isotis in the form of corm, corm, which is found in Colocasia, Gladiolus, Amorphophallus. Corm is used by some duck and other birds. Okay, other birds. Next point, previously Equisetum is used as previously equisetum is used by people in polishing wood furniture floor and of course dishes, alloy dishes for grinding alloy dishes. Next, different types of fans. Take part in different types of fans, different types of pteridophyte, especially fun pteridophyte of carbonic pteridophyte of carboniferous era, especially Cyathea. Example, Cyathea trifan, pteridophyte of carboniferous era took part in coal formation. Okay, coal formation. Number 8, the young stalks and stem of some pteridophyte like Marsillia teres are used as vegetable. Marsillia teres are used as vegetables. Okay, vegetables. Last of all, important use, that is starchy paste of Marsillia, sporocarp. Just a minute before I have told you what is sporocarp. Starchy paste of sporocarp of Marsillia, sporocarp of Marsillia is used in naddo cake by Australian people. Naddo cake by Australian people. Okay, so these are all about Pteridophyta. Thank you all.